do have a very exciting clinical trial that we're working on together. Excited about it because of the two drugs that are involved have a very interesting synergistic mechanism of action. And I've been helping out with Dr. Arriaga, who is the overall PI of the trial, and incorporating some interesting correlative imaging into the trial to better understand how these drugs are working together. So we are very excited about this trial, and so maybe I'll let her talk a little bit about the trial.
Sure. We've actually been working on it since its inception, so it's been a really great collaborative project. It is a phase 2 trial of a combination of drugs, one called cediranib, the other called olaparib, in recurrent glioblastoma. These are drugs, so in particular olaparib is a PARP inhibitor that has been shown to have activity in ovarian cancers, and in particular in BRCA-positive tumors. So PARP inhibition works by blocking an enzyme that repairs single-stranded breaks, and when you have a deficit in single-strand break repair and in double-strand break repair, the cell dies. So BRCA-positive tumors have this deficit in double-strand break repair, so when you add the PARP inhibition, then the hope is that the cell would die. We're combining it with cediranib because it appears that hypoxia, which is how this agent may work, will also decrease the repair of double-strand breaks. And because hypoxia is so important in this mechanism, we're incorporating imaging, which Dr. Gershon will talk about now, that can potentially look into which tumors are more hypoxic or how this drug is working.
So it's a really interesting opportunity for us to take these two very promising drugs and hopefully, help the care of patients, but also add in some additional correlative tests. And what's critically important is to identify those patients who are going to respond and understand why they're responding, and also understand why the population of patients that doesn't respond is not responding. And one of the ways we can do that is by looking at advanced imaging that really probes the tumor biology to let us understand longitudinally during the patient's treatment course what is happening to the tumor. How is the tumor responding to these novel agents? Is hypoxia changing? And so we have imaging techniques that allow us to measure these changes in tumor biology to better understand why these drugs are working or not, and better inform who might benefit from these drugs, what's the next step for patients, and how do we better move treatment forward for this challenging disease. Glioblastoma has certainly been in the press more recently, with famous politicians being diagnosed with glioblastoma recently. But if you actually look at the national statistics that are available, there hasn't been a change in the incidence of glioblastoma over the last several years for which we have statistics. On an individual community level of Massachusetts, those numbers are a little bit harder to track down, but at least from what we know for the national trends, there's not an increase in the number of cases of glioblastoma.
And I can add that in our clinical practice, I don't think that we have really seen any fluctuation in the numbers of patients who are diagnosed with glioblastoma. You also asked about the treatment, and that has really stayed fairly standard. So our standard of care therapy, which includes chemoradiation followed by adjuvant chemotherapy with temozolomide, has really been the standard of care and has stayed as such since 2005. There have been, other discoveries and potentially other things that may add to our armamentarium of how we treat the disease. There's been some work and some published data suggesting that the addition of other agents or actually devices could be considered, but really the standard of care has stayed the same.